Hello everyone, my name is Olaitan and I'm a PhD candidate at the University of Ibadan. Today I'm going to be talking about a COVID-19 study that I conducted earlier this year, and the title of my talk is Genomic Analysis Reveals the Abundance of SARS-CoV-2 Strains in COVID-19 Patients. Viruses are abundant microorganisms on Earth, and the novel coronavirus SARS-CoV-2 is seriously a global public health concern today. The disease that results from this viral infection is COVID-19, and over 12 million cases have now been globally confirmed. Certain strains of this virus may be life-threatening, thereby resulting in severe disease. With over 500,000 global fatalities resulting from SARS-CoV-2 infection, it has become critical to understand how this virus works and which strains are actively involved in COVID-19. Some of these viral strains might carry genes that encode proteins that modify host immune system in a way that results in higher disease severity, while some other strains might result in mild cases. The abundance of certain strains in patients might be potential diagnostic markers of disease outcome if the strains are unique in terms of clinical outcomes in infected hosts. I was therefore interested in the differential outcome after SARS-CoV-2 infection based on the biomarkers of what strains of the virus are in the sequences obtained from human patients. So I'm going to talk about the workflow of this study. RNA-seq data from oropharyngeal swab in human patients were obtained from the sequence read archive. And in order to ensure an improved assembly of repetitive regions, the library layout used for all of these data sets is paired end. So these were all paired end RNA-seq data. Assemblies of SARS-CoV-2 strains were also obtained from RefSeq, and there were 447 of those SARS-CoV-2 strains used in this study. Magic Blast was then used to map all of the RNA-seq reads to the reference genome database of SARS-CoV-2. And that process created sequence alignment maps that were then converted to BAM files and then eventually to bed graphs. Then I used Omic Streamer, which is another tool I developed to do some statistical analysis on the bed graphs. And that created some output, which I'm going to share in the next couple slides. This is a radar chart showing the most prevalent SARS-CoV-2 strains in COVID-19 patients. And the accession ID for the most abundant of those strains are MT263449 and MT246461. And there are other strains as well. SARS-CoV-2 strains in patients who survived. So the analysis reveals the following significant SARS-CoV-2 isolates and their corresponding coverage in a COVID-19 patient that survived. So we have 8 specific strains, and these strains had at least 50% coverage of the assemblies in all of the patient samples analyzed in this study. In terms of strain frequency, the analysis also reveals the following viral strains which were abundant in at least 80% of all of the samples analyzed. So 98% of these samples had the strains with accession ID MT2634449 and MT259245. All of these strains were in at least 50% of the patient samples. There were some other strains with their corresponding percentages in all the samples that are also shown on this page. So I'm going to move to the conclusion. So in this analysis, the following strains were found in 94% of the symptomatic COVID-19 patients used in the study. And the first one is severe acute respiratory syndrome coronavirus 2 isolate SARS-CoV-2 with the accession ID MT263449. And the second strain is severe acute respiratory syndrome coronavirus 2 isolate with the accession ID MT259245. Thank you very much for your time and attention, and I'll be happy to take questions.